The first application we are going to discuss today is the flavor saver tomato. This was the first crop to be genetically modified and sold to the market back in 1994. The main characteristic of this tomato is that it can have a longer shelf life due to the suppression of an enzyme called polygalacturonase or PG. When the product was launched to the market 27 years ago, it was a commercial success. Nevertheless, in 1998, its sales began to decline, mainly due to the fact that a British biochemist told in television that some rats have been presenting several biological effects due to the consumption of a genetically modified potato, not because of the product of the introduced gene, but because of the genetic engineering process itself. Thus, concerns were raised on the whole genetic engineering processes in the food industry. Despite these affirmations being proven to be false, the sales of the flavor saver tomatoes were so low that they were no longer sold after early 1999. In the flavor saver tomato, the protein that wants to be suppressed is the PG, who has been found responsible for the dissolve of the cell wall pectin in the tomato, which is the reason why the tomato softens as it ripens. The genetic modification performed in the tomato was insertion of a reverse orientation copy of the gene, with the aim of suppressing or at least diminishing as much as possible the expression of the PG. Also, in order to be able to create the PG antisense tomato line, it is necessary the presence of a cannabis-resistant gene as a selectable marker. As mentioned before, the PG gene was isolated from the tomato and inserted by reverse orientation in order to avoid expression of the protein. And all this process was done by agrobacterial-mediated transformation. The plasmid used for this objective was PCGN 1416, which is the one that we can see here in the image. We can observe several important biobricks. For example, we can see that the plasmid has a T region since the process is an agrobacterial-mediated transformation. As well, we can see the cannabis-resistant gene and most importantly, the pgcDNA in the antisense orientation. Now, according to Colgene, which is the company that developed Flavor Saver tomato, the vegetable is almost indistinguishable among non-genetically engineered tomatoes, except for two main characteristics. The first is that since PG enzyme is being suppressed, the tomato has a longer shelf life due to the fact that the cell wall degrades slower than those of non-GMO tomatoes, and that the paste, which was canned and sold massively back between 1994 and 1999, was observed to be more viscous when compared to traditional tomatoes. And now here in this image, we can see a comparison between non-GMO tomatoes and the flavor saver tomato. As we can see, the differences are quite impressive. As early as 20 days, we can see that the traditional tomato has already ringed a lot, and by 45 days, it is in an inedible state. But if we see that the flavor savory tomato, we can see that neither in 20 nor 45 days, the tomato looks in bad condition. Now we're going to talk about another application. This application is the transgenic virus-resistant papaya. This is produced in the United States, in the state of Hawaii. This state is one of the main producers of papaya, especially a region called Puna. 29 years ago, in 1992, it was discovered that a virus that has been affecting the crops of papaya. And this virus was the papaya ring spot virus, PRSV. In this application, the pathogen-derived resistance was used, meaning that the resistance is obtained when the genetic material of the pathogen itself. The process of resistance was done with biolistics by inserting a coat protein of a mutant of PRSV in embryogenic cultures of a type of papaya called sunset. And here in the image, we can see how the virus affects the papaya, giving it some rings on its peel. And as an important result of the genetic modification is that the resistance to the PRSV is durable, at least in Hawaii. Due to this, it was necessary to set up experiments that show if the papaya is resistant to only the Hawaiian strain of PRSV or also to other strains of PRSV. The experiment showed that the papaya was resistant to the Hawaiian PRSV strain, but susceptible to the PRSV strains with origin in Asian countries such as Taiwan, Thailand, or Guam. Also, in further studies, it was found that the pathogen-derived resistance is caused by the presence of interference RNA, which silences some genes. And we can see here in the image a healthy papaya crop in Hawaii, which were more commonly seen starting 1999, because from 1992 to 1998, the PRSB was severely affecting the papaya crops all over Hawaii. And while since the genetic modification of the papaya lies on the resistance of a virus, and viruses, as we know, mutate frequently, it is necessary to monitor if the rainbow papaya crops still have the resistance to PRSB. And the genetic modification of the papaya crops helped to recover the production of papaya in the region of Puna in Hawaii. In fact, in 1998, when the virus was in its peak, the production of papaya was only of 26 million pounds. And 3 years later, when the resistance crops were already present, the production of papaya rose up to 40 million pounds. And well, as we can see here in this table, the production of papaya in 1992, the year the virus was first identified, was of 53 million pounds, only in Puna. And the two subsequent years, 1993, 1994, the production increased, having a peak of 55.5 million pounds in 1994. But in the following 5 years, a severe decrease was observed, having the lowest production in the year 1999 of only 25.6 million pounds of papaya, not even half of the production of 1994. In 2000, 2 years after the introduction of the transgenic seeds, the production started to grow again, not recovering to the levels present at the beginning of the 90s, but it was possible to have a peak production of 40.3 million pounds of papaya in the year 2001. And due to these reasons, many experts agree that the biotechnology and genetic engineering have played a key role in the production of papaya. And it may be possible that if genetic modification of papaya was not done, the papaya will not exist nowadays, or it will be very difficult to produce due to its susceptibility of the virus.
Now, I'm going to talk about the genetic modified salmon. The development of a genetically modified salmon is one of the advances in genetic engineering within the past 30 years. The aqua vintage salmon founder animal was generated in the 1989 by microengineering a recombinant deoxyribonucleic acid rDNA construct composed of an element from an ocean food antifreeze protein gene and a protein coding sequence from a Chinook salmon growth hormone gene into the fertilized egg of wild Atlantic salmon. This salmon, produced by Aqua Bounty Technologies, is a triploid, effectively sterile, female fish containing a rDNA construct called OPA-FP GHC2, which imparts a rapid growth phenotype, allowing populations of these animals to reach a common growth measure, a small size, more quickly than populations of comparator Atlantic salmon. This new food animal engineer was developed by combining DNA from three animals: the Atlantic salmon, the salmon salar, deep water ocean elput, Macrosoarces americanus, and Pacific Chinook salmon, Oncorhynchus sadwisha. It is important to mention that Pacific Chinook salmon are a different species from Atlantic salmon. In order to manufacture an animal that grows unnaturally fast, the genetic engineering causes overproduction of an insulin-like growth factor hormone. Therefore, the new animal purportedly grows to commercial size in half the time of conventional Atlantic salmon. As shown in the image, the growth hormone from Chinook salmon joins a promoter from an ocean put, an eel-put fish spliced into an Atlantic salmon DNA, and finally, the new growth hormone directs the gene to produce hormone all year round instead of only in the summer. Now, you may be wondering if genetic engineered salmon are not manufactured to be sterile. Well, the answer is yes. The genetic engineered salmon eggs are treated to be sterile, but these methods are only 95 to 97% effective. With hundreds of thousands of eggs in production, these inevitable failure rates result in thousands of fertile fish. The adult salmon that have been engineered to fertilize these eggs, broodstock, are not sterile and could mate with native salmon in the wild. On the other hand, it is important to mention that if genetic engineered salmon escape into the environment, they will threaten native species via predation or competition for limited food or space, transgenic contamination, and an increase in exotic disease and parasites. Once engineered organisms are let loose into the environment, it is impossible to recall or eliminate them. Unlike chemical pollution, GA contamination is a living pollution that can propagate itself over space and time via gene flow. Now, I will explain some of the pros and cons of the genetically modified salmon. Starting with pros. Genetic engineered salmon could help meet rising demand for fish. This genetic modified salmon, it's 25% less food, thus makes fish farming more profitable. And finally, it could reduce pressure on wild fish stocks. And now, on the other side, the cons are that fish could escape from farms and harm wild salmon populations. Science on long-term effects of genetic engineered salmon is limited, and the concerns about genetic modified fish could further upset the ocean's delicate ecology, causing an ecological disruption or other species extinction. Now, the image of the right shows both fish at 18 months of age. It can be seen that aqua advantage salmon can reach adult size in the 16 to 28 months instead of 35 months for regular Atlantic salmon. This transgenic salmon eat 25% less feed and are about 20% more efficient converting the food to flesh. Now, I'm going to talk about the alfalfa. USDA first deregulated herbicide-tolerant alfalfa in 2005. Alfalfa, a perennial herbaceous forage crop known as lucerne, extensively grown in the world, is utilized as hay, silage, and for pasture. It is valuable feed for many classes of livestock because of a large dry biomass yield, high protein, minerals, and vitamins content. Due to its ability for nitrogen fixation, alfalfa enriches the soil, reduces the need of fertilization, and supports the growth of other plants. Improvement of some genetic characters, like resistance to abiotic and biotic stress, protein quality, and digestibility, could significantly increase the yield and nutritional value of alfalfa. Agrobacterium-mediated gene transfer has been basically used for transformation of embryogenic alfalfa genotypes. Transformed alfalfa plants were obtained by particle bombardment of kali and pollen. Efficient transformation was also achieved through microinjection and electroporation. Now, talking about its herbicide resistance, it is interesting that most of the trials carried out with alfalfa are focused on resistance to herbicides and diseases, thus resulting that weed problems are one of the most serious and difficult to control. One of the most destructive weeds in many alfalfa seed fields is dodder, Cuscuta spp, an annual parasitic weed, which causes 20% reduction of forage yield and blocks the seed production. Genes conferring resistance to herbicides were among the first which had been identified and used for this purpose. The first of alpha transgenic plants expressing a gene of practical interest was the one called BAR, that encoded resistance to the herbicides glufosinate ammonium, were obtained by Agrobacterium TI plasmid mediated transformation and analyzed under greenhouse and field conditions. And in order to promote alfalfa herbicide tolerance, the processes of Agrobacterium tumefaciens mediated transformation were optimized. Two different bacterial strains. The first one is the Hw4 and Hw1, containing respectively binary vectors carrying BAR gene for contact non-selective herbicide BASTA and AHAS3R gene for selective herbicide spleen were used in these transformations. It results interesting how, like other crops, transgenic alfalfa can be also used as bioreactor for producing of industrial enzymes, an approach significantly cheaper and conventional in contrast to expenses for constructing new fermentation facilities. In the following chart, we can appreciate how the alfalfa herbicide tolerance has increased in recent years to the genetically modified plant.
One of the most important applications of genetic engineering in food is the golden rice. It is well known that rice is the main staple food for hundreds of millions of people. This product is generally consumed without the outer layers, pericarp, tegument, etc. And the main reason is to remove the oil-rich iron layer, which turns rancid on storage. As the result of this, the edible part of rice grains consists of the endosperm, filled with the starch granules and protein spotted, causing the lacking in several essential nutrients for the maintenance of health. Therefore, dependence on rice as the main staple food contributes to vitamin A deficiency in at least 26 countries in the world. Golden rice was designed to produce beta-carotene, which is a natural precursor of vitamin A in the edible part of the rice. The original plant is capable to produce beta-carotenes in the leaf, where the precursor is involved in the function of photosynthesis, but it does not produce beta-carotenes natural in the endosperm because it doesn't require the use of photosynthesis. This is due to the absence of three enzymes, which are phytotin synthase, phytotin desaturase, and lycopene cyclase. Initially, the scientists tried to introduce all these genes into immature rice embryos, step by step, individually by particle bombardment. But it didn't work very well, and therefore, agrobacterium mediated transformation of immature rice embryos was used, designed to install to complete B-carotene pathway into the rice endosperm in a single transformation effort. Three vectors were constructed. The first one is the PB19HPC, which combines the sequences for a plant phytotin synthase originating from Narcissus phaedonarcissus and the sequence coding for a bacterial phytosin desaturase originating from Erwinia erudobora. These two being placed under the control of the endosperm specific lutein. The second one was carrying the same enzyme as the first one, but with a lacking of the expression cassette for the selectable marker APH4. And the last one was carrying the sequence coding for the enzyme lycopene cyclase under gluten promoter control. Bananas resistant to Fusarium. Fusarium oxyporum is a fungus that causes a disease called Panama disease, which affects the roots of a banana plant. The spore of this fungus infects plant roots and colonize the xylem vessels, blocking the flow of water and nutrients, causing extensive necrosis and the plant death. The problematic is that the spore of this fungus lie dormant in the soil until a substantial banana plant in established. Banana crops also present a lack of genetic diversity due to their asexual propagation, make them super vulnerable to disease. Also, this fungus can be eliminated from soil by any chemical treatment, also present resistant to any fungicides, and there is no treatment to cure this disease. The solution was the genetic engineer research from the Queensland University of Technology in Australia are working in a project where the Cavendish variety was modified with the addition of RGI2 gene, obtaining a variety called Grand named Cavendish resistant to TR4. The first gene, CED9 anti-apoptosis, gene derived from the nematode Canorhabditis elegans, confers resistance to fox race 1 in transgenic ladyfingers bananas. The second gene, RGA2, isolate resistant gene analogous to, is a putative nucleotide binding and leucine-rich gene from a seedling of Musa acuminata, which resistant to TR4. They constructed both CAD9 and RGA2 derived transgene expression cassette, where CAD9 was under the control of the mice polybicutin promoter and RGA2 was under the control of the Agrobacterium nopalin synthase promoter. These cassettes were transformed separately into Cavendish cultivar Grand Name, making it resistant to this disease.
So now with genetically modified corn, corn is the most commonly grown crop, and most of its GMOs, corn is created to resist insect pests or tolerate herbicides. Bacillus thuringiensis (BT) corn is a GMO corn that produces protein that are toxic to certain insect pests, but not to human, pets, livestock, or other animals. These are the same types of proteins that organic farmers use to control insect pests, and they do not harm other beneficial insects such as ladybugs. GMO BT corn reduces the need for spraying insecticides while still preventing insect damage. While a lot of GMO corn goes into processed foods and drinks, most of it is used to feed livestock like cows and poultry like chickens. Corn is one of humankind's earliest innovations. It was domesticated 10,000 years ago when humans learned to cross-pollinate plants and slowly turned a scratchily nondescript grass called teosinte into plump, productive modern corn. Genetically modified crops have the fastest adoption rate of the new technology in global agriculture simply because farmers benefit directly from higher yields and lower production costs. Here are some benefits and risks. Benefits. BT crops were intended to help farmers reduce not only the impact of pests, but also the use of agrochemicals in modern crop productions. According to Leonard Gianesi, Senior Research Associate at the National Center of Food and Agriculture Policy, farmers who adopt GM crops make more money in tougher times. Indeed, insect and virus-resistant traits have already saved several industries. According to the recent International Council for Science (ICSU) review of GM crops, disease-resistant corn crops may have lower levels of mycotoxins, potentially carcinogenic compounds to humans. They result from fungal activity in insect-infested corn crops. With fewer insect holes in plant tissue, associated fungi are not able to invade and produce toxins. Here are the risks. As pollen and seeds move in the environment, they can transmit genetic traits to nearby crops or wild relatives. Many self-pollination crops, such as wheat, barley, and potatoes, have a lot of frequency of gene flow, but the more promiscuous, such as sugar beets and corns, merit great concern. Seeds are more likely to spread GM's sugar beets genes into wild relatives. And breed so intensely for hundreds of years, most crops cannot survive without human intervention. BT corn is a type of genetically modified organisms. Corn produced through biotechnology is being used in many familiar foods, including cornmeal and tortilla chips. Corn is used to make high fructose corn syrup, which is used as a sweetener in many foods, such as soft drinks and baked goods. The FDA, U.S. Food and Drug Administration, considers BT corn to be nutritionally equivalent to the traditional corn. To transform a plant into a GMO plant, the gene that produces a genetic trait of interest is identified and separated from the rest of the genetic material from a donor organism. In the case of BT corn, the donor organism is a naturally occurring soil bacterium. Bacillus thuringiensis, and the gene of interest produces a protein that kills Lepidoptera larvae, in particular European corn borer. This protein is called the Bt delta endotoxin. Growers use Bt corn as an alternative to spraying insecticides. So here's the vectors ZMIR13L. This was used in event MON 863 corn, which is resistant to coleopteran. This vector is necessary for the Bacillus thuringiensis CRY3BB1 gene, which is the one that prevents the pest in the corns.